Hey everyone, welcome back to the channel. In the last video, we talked about the structure of DNA, what it's made of, how it's built, and why it's the ultimate information storage system in biology. Today, we're diving into one of the most fascinating processes in all of science, DNA replication. How cells copy their genetic blueprint with incredible accuracy. So let's start with the basics. DNA replication is the process by which a cell makes an exact copy of its DNA before it divides. Think of it like making a backup of your hard drive, except this backup happens at the molecular level, millions of times over. Every time a cell divides, it needs to pass on a complete copy of its genetic material to the new cell. If replication didn't happen, or if it happened incorrectly, you'd end up with broken instructions, and that could lead to mutation or disease. Now, DNA replication is what we call semi-conservative. That means when DNA is copied, each new molecule contains one original strand and one newly made strand. Why is this important? Because it ensures that the genetic information stays consistent. There's always a template to guide the creation of the new strand, and that template is the existing DNA itself. Replication doesn't start randomly. It begins at a special site on the DNA called the origin of replication. In bacteria, there's typically just one origin, but in humans, we got thousands. That's because our genomes are way bigger. At the origin, an enzyme called helicase comes in and starts unwinding the double helix, breaking the hydrogen bonds between base pairs. This creates a kind of open bubble in the DNA called a replication fork, and that's where the action happens. Here's where it gets a bit technical, but stick with me, it's really cool. DNA strands run in opposite directions. One goes 5' to 3', and the other 3' to 5'. But here's the catch. DNA polymerase, the enzyme that builds the new strand, can only add new nucleotides in the 5' to 3' direction. So what happens? Well, the strand that runs 3' to 5', the one that DNA polymerase can follow smoothly, that's called the leading strand. It's synthesized continuously, straight toward the replication fork. But the other strand, the one running 5' to 3', can be copied in one continuous piece. That's where things get creative. This strand is copied in short chunks called Okazaki fragments, named after the Japanese scientists who discovered them. Each fragment starts with a short RNA primer laid down by an enzyme called primase. Then DNA polymerase comes in and extends that primer with new DNA. Eventually, another enzyme, DNA ligase, comes in and glues the fragments together, forming a complete continuous strand. Let's pause for a moment and go through the team of enzymes involved in DNA replication. It's like an assembly line at the molecular level. First we got helicase. It unwinds the DNA helix. Then single-strand binding proteins come in to keep the strands apart, preventing them from snapping back together. Next, primase lays down short RNA primers so DNA polymerase has a starting point. Then the star of the show, DNA polymerase, adds new nucleotides to build the new strand. And finally, DNA ligase seals the gaps between Okazaki fragments on the lagging strand. Here's something amazing. DNA polymerase is not just a construction enzyme, it's also a proofreader. As it builds the new strand, it checks each new base it adds to make sure it matches the template. If it makes a mistake, and it does, occasionally, it can actually backtrack, remove the wrong nucleotide, and fix it before moving on. This proofreading ability is a big reason why DNA replication is so accurate. Errors happen only once every 10 million bases or even less. DNA replication works a bit differently in bacteria versus humans. In bacteria, there is usually one circular chromosome and a single origin of replication. Replication proceeds bidirectionally around the circle and finishes in about 20 to 40 minutes. In eukaryotic cells like ours, chromosomes are linear and replication is way more complex. There are multiple origins of replication along each chromosome, and replication moves outward in both directions from each origin. This helps speed up the process. After all, you're copying billions of base pairs. So if chromosomes are linear, how does replication deal with the very ends? Turns out there is a challenge. DNA polymerase can't fully copy the end of the lagging strand. This is known as the end replication problem. To solve it, cells use an enzyme called telomerase. It extends the ends of chromosomes using a special RNA template, kind of like patching the edge of a blanket. This helps protect the ends of chromosomes, called telomeres, which naturally shorten over time. When telomeres get too short, cells stop dividing. That's one of the factors linked to aging and cancer. Replication doesn't happen randomly. It's tightly controlled as part of the cell cycle. It occurs during the S phase. S is for synthesis. Before the cell divides in mitosis, it has to double its DNA, and replication is just the first step in making sure the two new cells each get a full, identical set of instructions. Why does all this matter in a real lab setting? Well, a lot of molecular tests, like PCR, which stands for polymerase chain reaction, are based on the principles of DNA replication. In fact, PCR is basically artificial DNA replication in a test tube. It uses DNA polymerase, primers, and repeated heating and cooling cycles to amplify a specific DNA segment. We use PCR in COVID testing, in genetic mutation analysis, in forensic science, and even in ancestry testing. What happens when replication goes wrong? Sometimes a wrong base gets inserted and isn't corrected. That's called a point mutation. Other times, entire sections might get duplicated, deleted, or inserted in the wrong place. These kinds of errors can lead to serious genetic conditions, or they can be harmless. It all depends on where in the genome it happens. That's why replication fidelity is such a big deal. One small error in the wrong gene can lead to disease like cancer, cystic fibrosis, or sickle cell anemia. Let's take a quick breath and review what we've covered. DNA replication is semi-conservative, one old strand and one new strand. It starts at origins of replication and moves outward. The leading strand is made continuously and the lagging strand in Okazaki fragments. A team of enzymes, helicase, primase, DNA polymerase, and ligase do the work. DNA polymerase has proofreading ability for high fidelity. In eukaryotes, replication involves multiple origins and telomeres. It happens during the S phase of the cell cycle. Mistakes in replication can lead to mutations, and that's why understanding this process matters. That's it for today's video on DNA replication. Pretty wild how our cells do all this automatically every time we grow, heal, or just live, right? In our next video, we'll look at enzymes that modify DNA, like restriction enzymes, ligases, and helicases, and how we use them in the lab to manipulate DNA for research and diagnostics. If you enjoyed this, please like the video, subscribe to the channel, and leave a comment if you have questions or want me to cover anything in more detail. Thanks for watching, and I'll see you in the next one.